Welcome everyone to your own channel, ADP for Learning. I am your own learning partner cum host, Ashish. So, let's begin our learning journey. Today, we are continuing our learning journey about the Interactive Glossary of Genomic and Genetic terms. And our motto is, 5 terms at a time, keep the learning in the mind. The first term for today is DNA fingerprinting. DNA fingerprinting is a laboratory technique used to determine the probable identity of a person based on the nucleotide sequences of certain regions of human DNA that are unique to individuals. DNA fingerprinting is used in a variety of situations, such as criminal investigations, other forensic purposes, and paternity testing. In these situations, one aims to match 2 DNA fingerprints with one another, such as a DNA sample from a known person and one from an unknown person. I guess a lot of people are first introduced to DNA fingerprinting while watching crime shows. An officer collects some samples from the crime scene. They put it in a tube. And then an hour later, they hold up a brightly colored gel, squint at it, and say, "Aha, we have a match for the killer's DNA." Then the show is over. Of course, that isn't exactly how things work in real life. But DNA fingerprinting is an important part of forensic science. Although it can't really tell you exactly who committed a crime, it can be used to help narrow down a list of suspects based on how well their DNA matches the samples that were found at the crime scene. Investigators can also use the DNA results to search specific databases to find other potential suspects. The second most commonly used term is DNA replication. DNA replication is the process by which the genome's DNA is copied in cells. Before a cell divides, it must first copy or replicate its entire genome so that each resulting daughter cell ends up with its own complete genome. DNA replication is probably one of the most amazing tricks that DNA does. If you think about it, each cell contains all of the DNA you need to make the other cells. And we start out from a single cell and we end up with trillions of cells. And during that process of cell division, all of the information in a cell has to be copied, and it has to be copied perfectly. And so DNA is a molecule that can be replicated to make almost perfect copies of itself. Which is all the more amazing considering that there are almost 3 billion base pairs of DNA to be copied. And replication uses DNA polymerases, which are molecules specifically dedicated to just copying DNA. Replicating all of the DNA in a single human cell takes several hours of just pure copying time. At the end of this process, once the DNA is all replicated, the cell actually has twice the amount of DNA that it needs, and the cell can then divide and parcel this DNA into the daughter cell, so that the daughter cell and the parental cell in many cases are absolutely genetically identical. The third popular term is DNA sequencing. DNA sequencing refers to the general laboratory technique for determining the exact sequence of nucleotides, or bases, in a DNA molecule. The sequence of the bases often referred to by the first letters of their chemical names, A, T, C, and G encodes the biological information that cells use to develop and operate. Establishing the sequence of DNA is key to understanding the function of genes and other parts of the genome. There are now several different methods available for DNA sequencing, each with its own characteristics, and the development of additional methods represents an active area of genomics research. DNA sequencing. In terms of information, DNA is like printed text. There is a method of storing the data or the printed word, a language to learn, and a process of understanding the meaning of what has been written. DNA sequencing is a process of reading. In the past, few people could read, most were illiterate. The expansion of literacy has meant that written information is accessible to a large group of people for interpretation, study, and communication. Advances in technology in our more recent history have had a similar effect on the human genome. Reading DNA through DNA sequencing has expanded access to the information in our genomes. The ongoing challenge going forward will be to expand understanding about what this information means for our health. The fourth popular term is dominant traits and alleles. Dominant, as related to genetics, refers to the relationship between an observed trait and the 2 inherited versions of a gene related to that trait. Individuals inherit 2 versions of each gene, known as alleles, from each parent. In the case of a dominant trait, only one copy of the dominant allele is required to express the trait. The effect of the other allele, or the recessive allele, is masked by the dominant allele. Typically, an individual who carries 2 copies of a dominant allele exhibits the same trait as those who carry only 1 copy. This contrasts to a recessive trait, which requires that both alleles be present to express the trait. Dominant refers to the inheritance of traits that are typically passed vertically from parent to child, where both the parent and the child are affected by the trait or disorder that is related to that gene. The most common form is autosomal dominant, where the associated gene is on one of the 22 non-sex chromosomes, and where the affected individual harbors two alleles of that gene, one pathogenic and one benign. In this inheritance pattern, harboring one pathogenic allele is sufficient to cause the person to have the trait. Then, an affected person having only one of two copies of the gene being pathogenic is what leads to the 50% chance of inheritance of the trait in their offspring. The fifth and the last term for today's video is double helix. Double helix, as related to genomics, is a term used to describe the physical structure of DNA. A DNA molecule is made up of two linked strands that wind around each other to resemble a twisted ladder in a helix-like shape. Each strand has a backbone made of alternating sugar, deoxyribose, and phosphate groups. Attached to each sugar is one of four bases: adenine or A, cytosine or C, guanine or G, or thymine or T. The two strands are connected by chemical bonds between the bases. Adenine bonds with thymine, and cytosine bonds with guanine. The discovery of DNA's double helical structure in the 1950s was perhaps the most significant biological accomplishment of the 20th century. Knowledge of this remarkably clever structure, involving two complementary strands of DNA that each provide the template for making the other strand, provided a key insight about how it was that DNA could serve as the information molecule of all living systems. This structural detail about DNA rapidly accelerated research that revealed important aspects about DNA function encodes information for creating and operating living systems. Meanwhile, DNA's double helix has arguably become the most well-known and iconic image associated with biology, perhaps with all of science. Thank you for joining with me for the amazing journey of learning. In the future, we will again meet with other new, innovative topics. Meanwhile, don't forget to like, share, subscribe, and comment.